Hi, everyone. My name is Rafer Cooley. I'm a PhD student in the Cedar Lab, currently an intern at Idaho National Labs, and my poster is titled Predicting and Detecting Future Malware Variants. This poster describes work that I'm doing for my dissertation, but I've been working with Taylor McCampbell on many aspects of the supporting work for this project. This project focuses on using artificial immune systems to generate malware indicators that are resistant to common obfuscation techniques. But first, a little bit of background on the main concepts related to this project. This project involves bio-inspired evolutionary algorithms, reverse engineering of malware, natural language models, and code clone detection algorithms. So after our biological immune system encounters a virus or bacteria, it is capable of building a library of indicators that allow it to detect the same or slightly altered versions of those pathogens so that it can mount a more effective response in the future. And the field of biological immunology has inspired a field of computer science that replicates those immune system principles digitally, which resulted in the development of artificial immune systems. One specific algorithm, called the clonal selection algorithm, attempts to mimic the process where B or T cells that recognize pathogens that have entered the body and are then reproduced to generate a population of similar cells in order to fight the infection. And eventually, some of those cells move on as memory cells to detect future infections. Malware authors have a history of evading antivirus detection through different obfuscation methods. They can add null operations that do not affect the end result of the program. They can split strings into long, complicated generator functions, or encrypt the main body of the program with different cryptographic schemes. One of the tools used to analyze malware is the Ghidra reverse engineering program from the National Security Agency. This program is capable of analyzing malware samples that are targeted for many different CPUs and operating systems, and it does this partially by converting the machine language instructions from the malware sample into an intermediate representation called P-code. Modern natural language models, such as GPT-3 and BERT, are capable of generating mostly coherent sentences and paragraphs of text from a simple input prompt. These models are making waves in the world of writing and content generation today, but the principles behind them have been applied to source code generation as well, such as GitHub's Copilot program and Microsoft's CodeBERT model. And the field of finding and detecting similar computer code is used by production software development companies, professors running programming courses, and cybersecurity analysts to determine how similar two programs are to one another. One method used for detecting similar code is fuzzy hashing, which works by hashing binary data in such a way that similar chunks of data will be evident in the resulting indicators. So now that we have a little bit of background on the different concepts we're using, we wanted to build this project around the question of, if given the first n samples of a piece of malware, where n is greater than 1, is it possible to generate a library of indicators that will successfully detect X% of future variants? Here we define future variants as any sample of malware detected after the first detection dates of our original n samples of malware. The detection date metric is a hard metric to work against, as those dates don't necessarily represent the dates that the malware variants were created or deployed, but it's a close enough metric for us to work with this project. Here we describe the methods of this project, where all the work done this summer is depicted on top of the gray background. With the help of our malware database project, we're able to feed malware samples through a bulk extractor that uses Ghidra to convert malware machine language instructions into the P-code intermediate representation language, and store the instruction sequences for each function of a malware sample into our database. Then we worked to develop multi-class classifier models by training the models on a sliding window view of the function instruction sequences in order to predict the instruction that would occur next after any given sliding window. Using an example dataset of DarkSide, Revil, WannaCry, and TrickBoot ransomware samples, we used a multi-class ADA-boosted decision tree model as our classifier and received satisfactory results around 75% F1 and recall scores. However, since we will be using these models for generating new instruction sequences, lower scores such as what we achieved will be beneficial. And this is because the new instruction sequences that they generate will be sufficiently different than the original sequences, which is what will be returned if we had reached 100% accuracy with our models. A challenge facing this project is the amount of time needed for Ghidra to analyze a single malware binary, which is quite high and could become cumbersome when we attempt to scale our research to the 6 million samples we have collected. And some points of future work include modifying our basic unit of data from being a sequence of instructions representing a function to being a data flow or instruction flow graphs, but working with graph data can be computationally expensive. Also, the decision tree classifier was an interesting proof of concept, but we would like to utilize the natural language models we spoke of earlier for our indicator generation process. And that's my project. Thanks for stopping by my poster.